Hello everyone, welcome to the next lecture in our physiology video lecture series. This lecture is titled Endocrinology. So let us start off by looking into what does endocrine exactly mean. We know that cell signaling between two cells can be of four different types. The first type is known as autocrine signaling, in which a cell A releases some factors which into the extracellular environment whose receptors are present on the cell A itself. So the factors released by cell A act on the cell A back. This is known as autocrine signaling. The second type of signaling is juxtacrine signaling. In juxtacrine signaling, the cell A releases various factors which act on a cell B that is directly very close to the cell A or attached to the cell A. This is known as juxtacrine signaling. Third is paracrine signaling. In paracrine signaling, the cell releases factors or hormones that affect only its local environment. And finally, we have endocrine signaling via endocrine glands present in various parts of our body, which release hormones right into the bloodstream, which can affect cells which are very far away from the source of the hormonal signal. This is known as endocrine signaling. The endocrine signaling is closely associated with the hormonal system, and in this particular lecture and the next one as well, we will look into how exactly the endocrine system works and how the hormones are produced and affect the target cells. So let us begin. First, let us have an introduction to endocrinology. The multiple activities of cells, tissues, organs of the body are coordinated by the interplay of several types of chemical messenger systems. These include first the neurotransmitters that are released by axon terminals of neurons into the synaptic junctions and act locally to control nerve cell function. Next are the endocrine hormones, which are released by glands or specialized cells into the circulating blood and influence the function of cells at another location, which can be far away from the source in the body. Neuroendocrine hormones are secreted by neurons into the circulating blood and influence the function of cells at another location in the body. Paracrines are secreted by cells into the extracellular fluid and affect neighboring cells of a different type. Autocrines are secreted by cells into the extracellular fluid and affect the function of the same cells that produce them by binding to the cell surface receptors. And finally, we have cytokines, which are peptides secreted by cells into the extracellular fluid and can function as autocrines, paracrines, or endocrine hormones. Examples of cytokines include interleukins and other lymphokines that are secreted by helper T cells and act on other cells of the immune system. So if you refer back to the lecture on immune system, you will understand that we have discussed in detail the activity of lymphokines and how the lymphokines secreted by the helper T cells help the entire immune system to be primed towards fighting a particular foreign organism or an infection. Next, cytokine hormones such as leptin produced by adipocytes are sometimes called adipokines. And thus we have numerous hormones that are produced or numerous factors that are produced that can take part in cell signaling. But in this particular lecture, we will look closely into the endocrine hormones and how their structure, function, and effect on the target cells. Next, let us look into the chemical structure and synthesis of hormones. In the schematic given here, we have the various endocrine glands that are present in our body that release these hormones. These include the hypothalamus, the pineal and the pituitary gland present in the brain, the thyroid gland, the parathyroid gland, uh, which are present in the thoracic region, the thymus gland. Stomach can also release hormones. The adrenal gland, which is one of the most important ones, pancreas and kidneys, the adipose tissue that is present beside the pancreas, the kidneys, the small intestine, and the gonads. One of the most important of all these glands is the pituitary gland, which is known as the master regulator of the endocrine system. And the hormones released by the pituitary gland control the rate of release of hormones by other glands. So let us first look into the chemical structure and synthesis of hormones. There are exactly three general classes of hormones. They include proteins and polypeptides, steroids, and derivatives of the amino acid tyrosine. Protein and polypeptides, including hormones secreted by the anterior and the posterior pituitary gland, the pancreas, which releases insulin and glucagon, the parathyroid gland, which releases the parathyroid hormone, and many others. Next, we have the steroids. Steroids secreted by the adrenal cortex, which include cortisol and aldosterone, the ovaries, which are which release estrogen and progesterone, the testes, which secrete testosterone, and the placenta, which releases estrogen and progesterone. Finally, we have the derivatives of amino acid tyrosine secreted by the thyroid, which include the hormones thyroxine and triiodothyronine, and the adrenal medulla, which release epinephrine and norepinephrine. These are the major classes of hormones, and there are no known polysaccharide or nucleic acid hormones. Next, we'll look into how exactly the hormones are formed and how they are released into the bloodstream. First, we will look into the polypeptide and protein hormones, which are stored in secretory vesicles. Most of the hormones in the body are polypeptide and protein hormones. These hormones range in size from small peptides, such as few as 3 amino acid, which is whose example is the thyrotropin releasing hormone, to proteins with almost 200 amino acids, such as the growth hormone and prolactin. Protein and peptide hormones are synthesized on the rough end of the endoplasmic reticulum, like most of the proteins as shown in the figure right here. The protein hormones are usually synthesized first as larger hormones that are not biologically active, known as pre-prohormones, which are then cleaved to form smaller prohormones in the endoplasmic reticulum. Prohormones are also not biologically active. So in this particular schematic, we are shown a signaling pathway which helps in the release of the particular hormones. In response to a stimulus, there is an activation of the secondary signaling system, which acts on the cell and activates transcription of the hormonal genes. The transcribed mRNA is then translated, and then the translated protein enters the endoplasmic reticulum for various folding and post-translational modifications. The translated protein is the preprohormone. As the protein is folding and undergoing various post-translational modification, it is cleaved into a smaller form, which is the prohormone, and via the Golgi apparatus and secretory vesicles released into the extracellular fluid. The prohormones are then transferred to the Golgi apparatus for packaging into the secretory vesicles. In this process, enzymes in the vesicles cleave the prohormones to produce smaller, biologically active hormones and inactive fragments. The vesicles are stored within the cytoplasm and many are bound to the cell membrane until the secretion is needed. Secretion of hormones as well as the inactive fragments occur when the secretory vesicles fuse with the cell membrane and the granular contents are extruded into the interstitial fluid or directly into the bloodstream via the process of exocytosis. So the take-home message from these slides is that the protein and peptide hormones are made in first in a biologically inactive form, which is the pre-pro hormone, which gets cleaved while undergoing post-translational modification in the endoplasmic reticulum, followed by followed by formation of the prohormone by the cleavage. This prohormone then is transferred to the Golgi apparatus, where it undergoes a packaging into secretory vesicles. The secretory vesicles are stored inside the cytoplasm or are in contact with the cell membrane and require an external stimulus to the cell in order to be secreted to the extracellular fluid via the process of exocytosis. Exocytosis is also a process that we have already discussed before, and I would recommend you to look back to the second lecture on Introduction to Cellular Physiology in order to understand the process of exocytosis. Next, we look at the steroid hormones. The steroid hormones are usually synthesized directly from cholesterol and are not stored in secretory vesicles like the protein hormones. The chemical structure of steroid hormones is similar to that of cholesterol, and in most instances, they are synthesized directly from cholesterol itself. They are lipid soluble and consist of three cyclohexyl rings and one cyclopentyne ring combined into a single structure as shown in the figure. So these one in the for the example we have here cortisol, which has this 1, 2, 3 cyclohexyl ring connected to this cyclopentyl ring right here. Although there is usually very little hormone storage in steroid producing endocrine cells, large stores of cholesterol esters in the cytoplasm vacuoles can be rapidly modified for steroid synthesis after a stimulus. Much of the cholesterol in the steroid producing cells comes from the plasma, but there is also de novo synthesis of cholesterol in steroid producing cells. Because the steroids are highly lipid soluble, once they are synthesized, they simply diffuse across the cell membrane and enter the interstitial fluid and then the blood. Thus, in case of steroid hormones, there is no need of any secretory vesicles to be formed. Now there comes the question why steroid hormones are not stored and are synthesized directly from cholesterol. One of the reasons behind this can be that storage of steroid hormones is difficult in the form because in any form other than cholesterol, because once biologically active steroid hormones are formed, they can easily diffuse across the cell membrane without any external stimulus. Thus, the cell cannot control the release of the steroid hormones easily. Hence, it is easier to directly make the hormone and allow it to normally diffuse out instead of storing the hormone and risk it being released when there is no reason for it to be released via external stimulus. Thus, steroid hormones are not usually stored while the peptide or protein hormones are. Next, we move on to amine hormones, which are derived from tyrosine. The two groups of hormones derived from tyrosine, which include the thyroid and the adrenal medullary hormones, are formed by the actions of enzymes in the cytoplasmic compartments of the glandular cells. The thyroid hormones are synthesized and stored in the thyroid gland and incorporated into macromolecules of the protein called thyroglobulin, which is stored in large follicles within the thyroid gland. Hormone secretion occurs when the amines are split from thyroglobulin, and the free hormones are then released into the bloodstream. After entering the blood, most of the thyroid hormones combine with plasma proteins, especially the thyroxine binding globulin, which slowly releases the hormones to the target tissues. Epinephrine and norepinephrine are formed in the adrenal medulla, which normally secretes about 4 times more epinephrine than norepinephrine. Catecholamines are taken up into preformed vesicles and stored until secreted. Similar to protein hormones stored in secretory granules, catecholamines are released from adrenal medullary cells by exocytosis. Thus, in case of amine hormones, we see both secretory granules storing the hormones in case of catecholamines, or peptide hormones such as the thyroglobulin. This peptide, the amine hormone, which is which are derived from tyrosine, which are stored by incorporation into the macromolecule protein thyroglobulin. Thyroglobulin itself cannot be released from the thyroid gland because it is too large to pass through it, and thus only when cleaved into smaller biologically active components, the hormone can be released into the bloodstream, where it is picked up by the thyroxine binding globulin and other plasma proteins to be transferred to the region where it acts. Next, we look into hormone secretion, transport, and clearance from the blood. First, the onset of hormone secretion after a stimulus and duration of action of different hormones. Some hormones, such as norepinephrine and epinephrine, are secreted within seconds after the gland is stimulated, and they may develop full action within another few seconds to minutes. The action of other hormones, such as thyroxine or growth hormone, may require months for full effect. Thus, each of the different hormones has its own characteristic onset and duration of action, each tailored to perform its specific control function. Next, we look into concentrations of hormones in the circulating blood and hormonal secretion rates. The concentration of hormones required to control most metabolic and endocrine functions are incredibly small. Their concentration in the blood range from as low as 1 picogram, which is 1 millionth of a gram in each milliliter of blood, up to a few micrograms. which is a few millionths of a gram per milliliter of blood. Similarly, the rates of secretion of various hormones are extremely small, usually measured in micrograms or milligrams per day. Next, we look into the transport of hormones in blood. In case of water-soluble hormones, such as peptides and catecholamines, they are dissolved in the plasma and transported from their sites of synthesis to the target cells or tissues, where they diffuse out of the capillaries into the interstitial fluid and ultimately to the target cells. Whereas in case of steroid and thyroid hormones, in contrast, circulate in the blood mainly bound to the plasma proteins. Usually less than 10% of steroid or thyroid hormones in the plasma exist free in solution. For example, more than 99% of the thyroxine in the blood is bound to plasma proteins. However, protein-bound hormones cannot easily diffuse across capillaries and gain access to their target cells, and therefore biologically inactive until they dissociate from the plasma proteins. The relatively large amounts of hormones bound to proteins serve as reservoirs, replenishing the concentrations of free hormones when they are bound to the target receptors or lost from circulation. Binding of hormones to plasma proteins greatly slow their clearance from the plasma. Next, we look into clearance of hormones from the blood. Two factors can increase or decrease the concentration of a hormone in the blood. One of these is the rate of hormone secretion into the blood. The second is the rate of removal of the hormone from the blood, which is called metabolic clearance rate. This is usually expressed in terms of number of milliliters of plasma cleared of the hormone per minute. In order to calculate this clearance rate, we need to measure the following: the rate of disappearance of the hormone from the plasma per minute and the concentration of hormone in each milliliter of the plasma. Then the metabolic clearance rate is calculated as follows. Metabolic clearance rate equals the rate of disappearance of the hormone from the plasma upon concentration of hormone in each milliliter of the plasma. And thus we can calculate the metabolic clearance rate using this particular formula. Next, we move on to mechanisms of hormone clearance. Like in the last slide, we discussed how metabolic clearance rate can be calculated by calculating the rate of disappearance of hormone and the concentration of the hormone. But how exactly does the hormone disappear or gets cleared from our system? The hormones are cleared from plasma in several ways, which include metabolic destruction by the tissues, binding with the tissues, excretion by liver into the bile, and excretion by the kidneys into the urine. For certain hormones, a decreased metabolic clearance rate may cause an excessively high concentration of the hormone in the circulating body fluids. For instance, this occurs for several of the steroid hormones when the liver is diseased because these hormones are conjugated mainly in the liver and then cleared into the bile. Next, hormones are sometimes degraded at the target cells by enzymatic processes that cause endocytosis of the cell membrane hormone receptor complex, wherein the hormone is then metabolized in the cell and the receptors are usually recycled back to the cell membrane. Or most of the peptide hormones and catecholamines are water soluble and circulate freely in the blood. They are usually degraded by enzymes in the blood and tissues and rapidly excreted by the kidneys and the liver, thus remaining in the blood for only a short time. For example, the half-life of angiotensin II circulating in the blood is less than 1 minute. Hormones that are bound to plasma proteins are cleared from the blood at much slower rates and may remain in circulation for several hours or even days. The half-life of adrenal steroids in the circulation, for example, ranges between 20 and 100 minutes, whereas the half-life of protein-bound thyroid hormones may be as long as 1 to 6 days. This was how exactly hormones are cleared from the metabolic system and then cleared out of the blood plasma. Next, we look into mechanisms of action of hormones. First, let us look into hormone receptors and their activation. The first step of a hormone's action is to bind to specific receptors at the target cell. Cells that lack receptors for the hormones do not respond. Receptors for some hormones are located on the target cell membrane, whereas other hormone receptors are located in the cytoplasm or the nucleus. When the hormone combines with its receptor, this usually initiates a cascade of reactions in the cell, with each stage becoming more powerfully activated so that even a small concentration of hormone can have a large effect. Hormonal receptors are large proteins, and each cell that is stimulated usually has some 2000 to 200,000 receptors. Also, each receptor is highly specific for a single hormone. This determines the type of hormone that will act on a particular tissue. The target tissues that are affected by a hormone are those that contain its specific receptors. The locations of the different types of hormone receptors are generally the following. This can include in or on the surface of the cell membrane. The membrane receptors are specific mostly for the protein peptides and catecholamine hormones. In the cell cytoplasm, the primary receptors for different steroid hormones are mainly found in the cytoplasm. This is mainly due to the fact that steroid hormones can pass right through the cell membrane and thus the receptors are present in the cytoplasm where once the steroid hormones pass through the cell membrane can bind to them. And finally, we have receptors that are present in the cell nucleus. The receptors for the thyroid hormones are found in the nucleus and are believed to be located in direct association with one or more of the chromosomes. Next, let us move on to intracellular signaling after hormone receptor activation. In this particular section, we will now look in detail into how exactly the various receptors and the hormones interact in order to produce the cellular action after the signal is received. First is the ion channel link receptor. Virtually all neurotransmitter substances such as acetylcholine, norepinephrine combine with receptors in the postsynaptic membrane. This almost always causes a change in the structure of the receptor, usually opening or closing a channel for one of their ions. This is something that we have discussed before when we have discussed the neuromuscular junction and various other synapses and looked into how exactly the nervous signals are communicated between neurons. The altered movement of these channels through these ions through the channels causes the subsequent effects on the postsynaptic cells. Although a few hormones may exert some of their actions through activation of ion channel receptors, most hormones that open or close ion channels do this indirectly, coupling with G protein linked or enzyme linked receptors. We'll now look into what exactly are the G protein linked hormone receptors in the next slide. So let us look into what the G protein linked hormone receptors are, or GPCRs are, which are also known as G protein coupled receptors. Many hormones activate receptors that indirectly regulate the activity of target proteins, for example, enzymes or ion channels. By coupling with groups of cell membrane proteins called the heterotrimeric GTP binding proteins or G proteins. What does the heterotrimeric means? That the protein is made of three subunits. Hetero obviously means that the three subunits are different from each other. And the GTP binding protein is because they can bind to GTP, which is guanine triphosphate, which uh, which is guanine triphosphate, and can also act as a source of energy for the cell. There are more than 1,000 known G protein coupled receptors, all of which have 7 transmembrane segments that loop in and out of the cell membrane. Some parts of the receptor that protrude into the cell cytoplasm, which include especially the cytoplasmic tail of the receptor, are coupled to the G proteins that include three. That is, the protein is trimeric parts, the A, B, and G subunit, or we can say the alpha, beta, and gamma subunit. When the ligand or the hormone binds to the extracellular part of the receptor, a conformational change occurs in the receptor that activates the G protein and induces intracellular signals that either open or close the cell membrane ion channels or change the activity of an enzyme in the cytoplasm of the cell. The trimeric G proteins are named for their ability to bind to the guanosine nucleotides. In the inactive state, The A, B, and G, or the alpha, beta, and the gamma subunits of the G proteins form a complex that binds to guanosine diphosphate or GDP on the A subunit. When the receptor is activated, it undergoes a conformational change that causes the GDP-bound trimeric G protein to dissociate with the cytoplasmic part of the receptor and to exchange GDP for guanosine triphosphate, that is GTP. Displacement of GDP by GTP causes the A subunit to dissociate from the trimeric complex and to associate with other intracellular signaling proteins. These proteins in turn alter the activity of the ion channels or the intracellular enzyme. Here we have a schematic which shows us the mechanism of action of the GPCR. So here we have a receptor which is a 7 transmembrane protein which has a part of it that extrudes out of the cell membrane which accepts the which binds to a ligand, and this causes a change in the G protein that is coupled to the receptor. The G protein contains the alpha, beta, and gamma subunits. The beta and gamma subunits are bound to the cytoplasm. The alpha unit is bound to the beta and gamma. Alpha unit is also bound to a GDP moiety in the inactive state. But once the hormone binds to the 7 transmembrane receptor, there is a conformational change that occurs in the receptor which activates the G. Upon activation, the GDP bound to the to the alpha subunit of the G protein is exchanged for a GTP. This shows us this event is the activation of the G protein. Once the G protein is activated, the alpha unit now dissociates from the gamma and the beta unit. The gamma and beta unit are still bound in cytoplasm while the alpha unit is now free. The alpha unit now goes and binds to various other proteins or secondary messengers and and this event now produces a secondary signal that causes various other events downstream from the hormone and receptor binding. This is how the hormone binding to the receptor activates the G protein, and then a secondary messenger system is activated in order to prepare the cell to respond to the stimulus of the hormone. Next, we move on to enzyme-linked hormone receptors. Some receptors, when activated, function directly as enzymes or are closely associated with enzymes that they only activate. These enzyme-linked receptors are proteins that pass through the membrane only once, in contrast to the 7 transmembrane G protein coupled receptors. One example of an enzyme-linked receptor is the leptin receptor. The leptin receptor is a membrane of a large family of cytokine receptors that do not themselves contain enzymatic activity but signal to associated enzymes. In case of the leptin receptor, one of the signaling pathways occur through a tyrosine kinase of the Janus kinase family of the JAK family, which is known as JAK2. The activated JAK2 molecule then phosphorylate other tyrosine residues within the leptin receptor JAK2 complex to mediate intracellular signaling. The intracellular signaling includes the phosphorylation of signal transducer and activator of transcription protein or STAT protein, which activates transcription by leptin target genes in order to initiate protein synthesis. Here we have a schematic which shows the leptin receptor pathway. So first we have the leptin hormone bind the leptin receptors. This activates the JAK receptor present here, which in turn phosphorylates the STAT3 proteins. Phosphorylated STAT proteins then dimerize, which is the active form of the STAT proteins, and enter into the nucleus to begin transcription of various hormone downstream genes. This transcription of genes produce mRNA, which they are then translated to proteins, and hence we have physiological effects as a result of the leptin signal. There are other examples of enzyme-linked receptors such as the RTKs as well, which are receptor tyrosine kinases, which also act via phosphorylation and activation via hormone binding and phosphorylation of particular amino acid on the tyrosine groups, and which signal via the phosphorylation of various secondary messengers, and the secondary cascade is similar in which finally after for a repeated phosphorylation of various molecules, there is a transcription factor that is generated which enters the nucleus and begins transcription of hormone downstream genes. Next, move on to intracellular hormone receptors and activation of genes. Till now, we have been seeing receptors which are present right on the membrane of the particular cell. These receptors are mainly here for the peptide and the protein hormones, which obviously cannot pass through the cell membrane, and thus the receptors of them need to be on the cell surface themselves. But in case of steroid hormones, as we have seen, they can easily pass through the cell membrane, and thus the receptors of them are present inside the cytoplasm itself. This is the point that we will now discuss. Certain hormones, including adrenal and gonadal steroid hormones, thyroid hormones, retinoid hormones, and vitamin D, bind with protein receptors inside the cell rather than in the cell membrane. Because these hormones are lipid soluble, they readily cross the cell membrane and interact with receptors in the cytoplasm or nucleus. The activated hormone receptor complex then binds with specific regulatory promoter sequence of the DNA called the hormone response element. And in this manner, either activates or represses transcription of specific genes and formation of messenger RNA or mRNA. Thus, we have a schematic showing us the whole process. The lipophilic hormone diffuses right through the cell membrane and enters into the cytoplasm. From here, the hormone can attach to a cytoplasmic receptor, and the hormone and receptor complex can be translocated to the nucleus, or the hormone can directly enter the nucleus and attach to a nuclear receptor, forming the activated hormone receptor complex inside the nucleus itself. After the formation of the hormone receptor complex, it binds to a special region of the cell of the cells by it binds to a special regulatory sequence present in the nucleus, which is called the hormone responsive element. Once it binds to the hormone responsive elements, it activates or represses transcription of genes downstream to this particular element, and thus we see transcription of genes and formation of mRNA, which is then translocated out translocated outside the nucleus for the translation to proteins. And thus, after the formation of proteins, we see a physiologic change or a response of the cell to the particular hormone stimulus. Next, we have the second messenger mechanism for mediating intracellular hormonal function. One of the means by which hormones exert intracellular action is to stimulate formation of a secondary messenger or a second messenger. The second messengers can be the adenyl cyclase CAMP second messenger system, the calcium ions and associated calmodulin system, and products of membrane phospholipid breakdown. We'll go through all of these systems individually. First, we have the adenyl cyclase CAMP second messenger system. Binding of hormones with the receptor allows coupling of the receptor to a G protein. If the G protein stimulates the adenyl cyclase CMP system, it is called a GS protein, denoting it to be a stimulatory G protein. Stimulation of adenyl cyclase, a membrane-bound enzyme, by the GS protein then catalyzes the conversion of a small amount of cytoplasmic adenosine triphosphate or ATP into CAMP, which is the cyclic adenosine monophosphate inside the cell. This then activates CAMP dependent protein kinases, which phosphorylates specific proteins in the cell, triggering biomechanical reactions biochemical reactions that ultimately lead to cell's response to the hormone. Here we have a schematic representation showing us the whole process. First, we have the hormone binding to the receptor. The receptor changes its conformation, which in turn activates the G protein coupled to the receptor. The alpha-subunit dissociates from the gamma and beta and activates adenyl cyclase, which is a membrane-bound protein. Once adenyl cyclase is activated, it converts cytoplasmic adenosine triphosphate into cyclic adenosine monophosphate or CAM. The ATP, when ATP is present, we have inactive CAMP dependent protein kinases. But once CAMP is formed, it can activate various CAMP dependent protein kinases. And thus, the protein kinases in presence of CAMP phosphorylate various other downstream proteins, and these proteins, this phosphorylation cascade continues till we have a cellular response to the hormone. Thus, the adenyl cyclase CAMP second messenger system produces the cell's response to the hormone signal. One of the things that you have to keep in mind is why is this system known as the second messenger system. The first messenger in the whole scenario, if we keep in mind, is the hormone, which is received by the cell. The hormone is the first external signal that the cell receives, and conversion of its hormone signal to an intracellular signal is done by the second messenger, that is the three systems that we are now discussing. These second messengers are very important and are essential for the cascade to continue in order for there to be a proper cellular response. Next, we move on to the cell membrane phospholipid second messenger system. Some hormones activate transmembrane receptors that activate the enzyme phospholipase C. attached to the inside projections of the receptor. This enzyme catalyzes the breakdown of some phospholipids in the cell membrane, especially phosphatidylate inositol biphosphate, which is also known as PIP2, into two different second messenger products. Inositol triphosphate, which is IP3, and diacylglycerol, which is DAG. The IP3 mobilizes calcium ions from the mitochondria and the endoplasmic reticulum, and the calcium ions then have their own second messenger effects, such as smooth muscle contraction and changes in cell secretion. DAG, the other lipid second messenger, activates the enzyme protein kinase C or PKC, which then phosphorylates a large number of proteins, leading to the cell's response. So here we have the schematic which shows us how this particular cascade works. First, we have a peptide hormone binding the receptor. This activates the G protein, which in turn activates the phospholipid C. Phospholipid C breaks down PIP2 into IP3 and DAG. DAG goes on to activate the protein kinase C to its active form, which then phosphorylates various proteins. And thus we have phosphorylated proteins which start another cascade which lead to the cell's response to the hormone binding. IP3, on the other hand, acts on the endoplasmic reticulum and the mitochondria in order to release calcium. Calcium release has another starts another cascade, which again bring about separate cell responses. What exactly are the calcium dependent cell responses of our calcium dependent secondary messenger system is something we'll look into just now. Another second messenger system operates in response to entry of calcium into cells. Calcium entry may be initiated by changes in membrane potential that open calcium channels or hormone interacting with membrane receptors that open calcium channels. On entering a cell, calcium ions bind with the protein calmodulin. The calmodulin changes its shape and initiates multiple effects inside the cell, including activation or inhibition of protein kinases. Activation of calmodulin dependent protein kinases causes phosphorylation, activation, or inhibition of proteins involved in the cell's response to the hormone. For example, one of the functions of calmodulin is to activate myosin kinase, which acts directly on myosin of the smooth muscles to cause smooth muscle contraction. Myosin kinase and the calcium calmodulin system in order to initiate smooth muscle contraction is something we have discussed about in detail in previous lectures on muscle contraction and muscle um, muscle membrane potential. I would request you to refer back to those videos if you are having problem recalling those. And in this particular section, where we are looking at the calcium calmodulin second messenger system, calcium acts as the second messenger directly. That is, calcium entry into the cell is initiated as a result of the hormone binding to a particular receptor, thus making calcium the second messenger. But in case of the cell membrane phospholipid second messenger system, in which IP3 acts on the endoplasmic reticulum in order to release calcium, calcium is not the second messenger, but part of the downstream cascade of the phospholipid second messenger system. Next, we move on to hormones that act mainly on the genetic machinery of the cell. Steroid hormones increase protein synthesis. The sequence, the sequence of events in the steroid for hormone function is as follows. The first, the steroid hormone diffuses across the cell membrane and enters the cytoplasm of the cell, where it binds to a specific receptor protein. Combined receptor protein hormone complex then diffuses into or is transported into the nucleus. The combination binds at specific points on the DNA strands in the chromosome, which activates the transcription process of specific genes to form mRNA. The mRNA diffuses into the cytoplasm where it promotes the translation process at the ribosomes to form new proteins. This is the mechanism of action of how exactly steroid hormone function and bring about protein synthesis. Next, we look into the thyroid hormones and how exactly thyroid hormones increase gene transcription in the cell nucleus. The thyroid hormones, thyroxine and triiodothyronine, cause increased transcription by specific genes in the nucleus. To accomplish this, these hormones first bind directly with the receptor proteins in the nucleus itself. These receptors are probably protein molecules located within the chromosomal complex, and they likely control the function of the genetic promoters operation. Promoters are operated. Thus, the thyroxine and thyroid hormones cause increased transcription in the specific genes by binding to receptor proteins present in the nucleus itself. So, if we go back to various hormone receptors, we have seen that specific receptor to a special hormone, some hormones have their receptors present in the cytoplasm, and some have the receptors present in the nucleus itself. We have seen that steroid hormones have mostly the receptors present in the cytoplasm, and thyroid hormones have the receptors present in the nucleus. Next, we look into two important features of the thyroid hormone function. These features include, first, they activate the genetic mechanism for the formation of the enzymes that promote enhanced intracellular metabolic activity in virtually all cells of the body. And once bound to the intranuclear receptors, the thyroid hormones can continue to express their control function for days or even weeks. As we have seen before, that there are specific hormones that can act for longer periods of time, but also require longer periods of time to bring about the proper full effect on particular cells. One of the examples of these particular hormones include the thyroid hormone. Secondly, the thyroid hormone does not specifically produce the gene transcription to produce some protein or a particular protein. Thyroid hormones can act on several cells or almost all cells of the body in order to increase the metabolic rate of the body. And this is an important function that is associated with thyroid. So that was all we had for today. These are the references to the slide that we have discussed today. They include the chapter 74 from the textbook of medical physiology, RPC Guide, the 11th edition. I thank you for listening to this video.